No worries. So our story begins in 1961. Okay. Let me just. Okay. So there was a scientist working in Wisconsin in the middle of North America, and he was studying a corn fungus. Every growing season, Wisconsin's a very cold place, right? So in winter, it's completely covered by ice. No plants are growing, uh, but they grow a lot of corn. Every growing season, he would take leaves from last year that were infected by a particular corn fungus, grind them up, and put that powder on new corn plants that are growing. So I have a question for you, and raise your hands and I will call upon you. Why would he do this? Go ahead, Faris.
Sir, I think that he would put this powder on the new plants so then maybe he could find out why the corn plants were being infected and then he would find out a solution on how to treat this issue.
But he knows why they're getting infected. Like he's literally taking infected leaf powder and putting it on them.
Sir, he could be doing
Right?
it to track.
Srivardhan, hold on. We're just going with raised hands. Harshadit?
Sir, maybe he could be testing if this disease has an immunity within different corns.
Okay, do different individuals have different immunity to the disease? Krishana?
Maybe he wanted to see if the corn reacted differently to the fungus in different seasons. So maybe in summer the fungus would be worse off for the corns than in winter, and maybe they spread more easily in summer.
Okay, that's a good point. So one could be immunity, one could be over time. Different years could result in different outcomes. Srivardhan?
One could be that he wanted to check whether the disease is spreading into the seeds and therefore into the next crop of the same corn.
How does the disease spread? Okay, and what's the advantage of putting the disease himself for all of these things?
So he can track which plants have a disease, which don't, and he knows where the disease is.
Right. So if you were waiting for the disease to naturally come, suppose it was a question of different plants having different immunity, you don't know whether the plants that did not get the disease did not get it because it was immune or because the disease just didn't happen to come there. This is the fundamental point of an experiment. You control some things. And I'll make sure to get around to everyone. Shashwat and Prapanch, I see your raised hands, but we'll come there. Oh, okay. Wait. Yeah, we can actually do time for the raised hands. Shashwat.
Sir, I am saying that he did this to understand
Shashwat,
the
just sit
disease.
and answer. No need to stand. Don't worry.
He did this to understand the disease better, so it could eventually be controlled or prevented.
Right, but what would he understand from about the disease by putting it on fresh, poor, innocent plants?
Fungus and corns. That fungus infects corns.
Okay, think of like we're thinking of, because it's plants, we're like sure, but think about this in a human context. Someone is literally taking disease and going around and putting it on people.
Sir?
Yes, who is this?
I'm Aditya, so can I answer?
Yes.
He wanted to check whether the disease only affects corn or other plants, is my opinion.
Okay, corn or other plants. Okay, so it could be, but in order for him to test that, what would he need to do different from what I've written on the board, on the screen so far?
You would need to use other plants other than corn.
Right. So far, he's only been putting it on corn. He'd have to put it on other plants as well. Okay. So anyway, let's go to the next slide. In 1961, a surprise frost happened. Does anyone know what a frost is? when I'm saying a surprise frost happened?
It's when ice forms on plants and crops and doesn't allow them to grow.
Okay. And how does that happen all of a sudden?
Because the temperatures suddenly drop.
Right. So a frost is usually early in spring or late in autumn, right? Not winter time. The days are pretty warm, but you suddenly have a cold day. Okay, I think Abhinav can probably relate to this because he's from Norway. I mean, he's living in Norway. I lived in Minnesota for 5 years, northern part of the USA, and we saw plenty of frosts. And you get frosts in the Himalayas as well, right? You suddenly have a cold night and ice forms. Now, something strange happened that day. Only plants that were infected with the disease got damaged by the frost. But the healthy plants were not damaged by the frost. So first I want to ask you a question. Why is cold temperature damaging to plants? Prapanch?
Maybe it's because they don't have enough. I'm not sure. Maybe it's because they don't have enough heat.
They don't have enough heat. So why does that cause damage? Why is a lack of heat damaging?
Because they can't transpire.
They can't?
They can't do transpiration.
I'm sorry, you're very soft, not able to hear you.
And they might not be able to do transpiration or respiration.
They need to transpire. Okay. So here, the frost happened in the night. Are the plants very active in the night, Paris?
No.
Sir, I think that it was damaging because maybe because of the ice crystals, as we
So sharp, right? And so therefore they can poke things and hurt them. Then the question was, where is the ice coming from? Which nobody answered as such.
Sir.
So where is the ice coming from? Let me ask someone who hasn't spoken so far. Himank.
Yeah, so the ice could come from the surroundings.
Where in the surroundings?
It could come from either rain on the ground. It could be falling snow.
Okay, so why would that be dangerous? Why would snow, like, I've had snow fall on my hand. It's not really dangerous.
It's dangerous if you are not having proper clothing or if you stay out for a long time, because then you can feel hypothermic. Or if, say, for example, you're on a ice lake which has a thin layer of ice, you might fall through and drown.
I'm not commenting on that right now. So we all know that everything is made out of particles, right? You've all heard about atoms, molecules, this and that. I want you to think about it at a particle level and answer the question, how does water freeze? Like, what is actually happening when water freezes? Krishana, your hand is up.
So when water freezes, so water in the beginning is in its liquid state. So the particles are still, they're all touching, but they're all against each other, but they can move around. They're not moving around freely, but they're still moving around. When the temperature drops, they lose energy. So then they stop moving around and they become fixed in a certain place. And then they don't move around anymore. Instead, they start vibrating in a fixed position. So earlier they were all touching, but they were still moving around. But now they're all obviously still touching. There are very few gaps. They are held together more tightly, so the intermolecular forces become tighter, and they're fixed in one position and they're only vibrating. They're not moving around because they don't have any more energy since the temperature dropped and they lost energy because of that.
Okay, so one thing implicit in what you're saying is that temperature what we measure as temperature on our scale, when we look at the particle scale, that is movement of particles. Higher temperature means higher movement, lower temperature means lower movement. Okay. Does anyone have anything to add on top of this? Shashwat? Shashwat, you're not near.
Sir, now I am audible.
Yes.
So I can add that ice can form first outside the cells.
No. So this is just simple, regular. No, let's not worry about cells. This is just how does water freeze? Normal water in this glass.
Tiny particles like dust or bacteria or the disease powder can act as a starting point.
No. So this is in general. We're talking about how does water freeze in general. So far, what we've discussed is normal water is moving around very fast. But then once the temperature lowers, that motion lowers, then they sort of, does this kind of make sense that ice would look like this if you looked at it on the particle level? Dhruv? Dhruv, you're muted. You are still muted.
So like, am I audible?
Yeah.
Okay, so I think it'll be the opposite because actually solids
slightly positive and that slightly negative charge can attract and they can structure each other. Okay. Laksh, you have raised your hand. And you're muted, so please unmute yourself. I still can't hear you. Okay, so actually, while Laksh figures out his audio situation, I want to show you a small video, which will hopefully load. Wait. Hold on. Sorry, I need to go to YouTube. Hi, Kaka. Okay. So, here's the thing.
Yeah, I tried it out myself.
And now you can see clearly that the water is still liquid. You can see this air.
It's been in the freezer for 2 days, or like for plenty of time to freeze, but it hasn't frozen.
Sir, can you put it in full screen?
Air bubble floating around. And now I'm going to bang it on the counter and we will see if we can get an ice crystal to form. Whoa, look at that! You can see that ice crystal growing right down from the top to the bottom.
But I didn't quite get the transition.
Okay, let's just watch that transition again. So he had a bottle full of supercooled water. Okay. Now, first off, supercooled water. Water is supposed to freeze at 0 degrees Celsius. But here it is at -10 and nothing's happened.
See if we can get an ice crystal to form.
And all he did was hit it once.
Whoa, look at that! You can see that ice crystal growing right down from the top to the bottom. So there you have it. I was finally able to demonstrate the supercooled water.
Okay. So what's happening there? Anyone? Cool. So I'm going to hold over there. Otherwise, we'll never get through a story. And whoever is unmuting and speaking, please wait until you're called. So here's another video. But before we start the video, I want to point out to you. So this is a molecular dynamics simulation. It's how scientists study these things at the molecular level. And what's on the top?
A what simulation?
This is just so that you can find it on YouTube if you sorry.
It looks like a molecule, it looks like a compound probably, something which like combined together chemically.
Okay, if you want to find these kinds of things on YouTube, just search for molecular dynamics. That's what this kind of simulation is called. And so note that in each place over here, you've got this big red thing and two small yellow things. That's the same as oxygen and two hydrogens.
Hello, am I audible?
Yeah, so just pointing out over here, it's a little hard to see. But if you look at each of these, you can see that there is a big red thing and two small white things. The big red thing is the oxygen atom. The two small white things are the hydrogen atoms. And if you look at it carefully, you can see at every point, this is oxygen, two hydrogens, oxygen, two hydrogens. And there are gaps always, right? That's a gap, that's a gap, that's a gap. So what do you think this top part is and what is the bottom part?
Bottom is solid and top is liquid.
Who is that?
Me, Dhruv.
So me is not helpful because I can't see people over here. So yes, name, Dhruv. Indian? So Dhruv is saying the bottom is solid and the top is liquid.
Sir, actually, I think the bottom is actually liquid.
The bottom is liquid, the top is solid.
But
Because
people are saying the bottom is liquid and the top is solid.
bottom is
Darsh, why do you feel like, wait, hold on. Darsh, why do you feel like the bottom is liquid and the top is solid?
the top has a crystalline structure with a hexagonal pattern. Meanwhile, the bottom has an irregular structure.
Right, and we just discussed, remember we've been discussing that ice is crystals, right? Let me open this so that you can see it. How many times is it different? Right, so let's just watch this. Skip all that. This is from an Australian lab. Let's get rid of the horrible sound. So this is a simulation of water freezing into ice. What do you think is the temperature of this whole area?
Between 0 to 4 degrees.
Assembly in the morning at school, you can just raise your hand. You have assembly in the morning at school. But before assembly, you're all sort of milling around, right?
So basically, basically on the first day of school, the teacher just does like we all get our spots, and then the rest we know we memorize our spots, and we just go in and once the teachers tell us assembly, we just walk to our spots.
Okay, so when I was in school, things worked differently. Maybe we all weren't as intelligent and didn't remember our spots. The teacher was standing in front, and we knew that our class teacher is there, so our line starts from there. Where this mess over here, and from the front, a line starts forming. And because we can see the structure of the line, we're then able to join it. This is exactly what is happening when ice.
It's herd mentality kind of thing.
We have a lot of space between us.
The point is that for the ice to crystallize, it needs some structure to be able to start from. Suppose your teachers did not stand in front and you did not know your locations in the whole place. You're suddenly not going to collapse into a regular structure. But once there is a regular structure, the next person in the line can latch onto it, can latch onto it, can latch onto it. And when we're looking at this video, I'm actually just going to switch to the video briefly. You can see that at each of these points, it's not like a molecule directly slots in. There's a lot of shuffling around. And then suddenly one happens to fit in. Even after fitting in, it can sometimes be shuffled out. And the whole thing is already at 0 or below 0 degrees. Okay, now nobody answered the question yet. This good question, we're getting to it later. There's literally a slide on it. So, wait, it's about time that I introduce you to the hero of today. Okay, and Aditya, yes, I will come to you next. Okay, this is a rod-shaped bacterium called Pseudomonas syringae. It was isolated in the 1970s. Okay, so we're talking about 50 years ago. Okay, and it is one of the best known plant pathogens out there. Disease organism that infects plants is very, very well studied. Okay, so coming to the question of how good is it at making ice? One more video, which will again load. Okay, so okay, you can see it. So that is supercooled water, exactly like it was in that Veritasium video. And now they're just going to drop some of the bacteria in it. And instantly that vial has frozen. They just dropped a little bit of the bacteria in it. Okay. So I had been talking about the scientist who was researching on corn. There were other groups of scientists who were trying to understand what makes water freeze. Okay. And they found that clay is very, very bad at making water freeze. Okay. They found that soil is better at making water freeze. They tried a whole range of things, but they weren't able to get better than making water freeze at -5 degrees Celsius. Okay. So if you take completely pure water, it does not freeze. Because just like I said, so completely pure water doesn't freeze until about -30 degrees. Okay. If there is something for the water to condense around, like the teachers in front of the line that create the structure, then it can freeze at a much higher temperature. So these scientists tried different things, and the best that they could get to was -5 degrees Celsius. Pseudomonas syringae does it at -3. So it was better than everything else that the scientists could find. We'll get there. Okay? So a good chunk of the Earth's surface is frozen for good chunks of the year. So life has had to figure out how to deal with cold temperatures. Let me just get through my story. Okay, I know there are a lot of raised hands. So can anyone recognize this fruit over here?
Mango.
Mango? Any other guesses?
Looks like coconut.
Coconut. So Pseudomonas is a huge problem for global agriculture. The numbers are on the next slide. Oh, well, I didn't make my images well. So there are several images over here. I'm sorry, I stacked them differently. You will see images of Pseudomonas. If I'd stacked them properly, you'd see images of Pseudomonas infecting cherries as well. Okay. It infects soybeans, beans, kiwis, apples, lots of things. Right? In New Zealand, this infection dropped orchard values by almost like 7-fold. Right? An orchard that was giving $450,000 of produce ended up per hectare gave only $70,000 per hectare. Okay.
Just because of one infection. Because
of this bacterium. Different varieties of it, but it's all Pseudomonas syringae. So this is why it is one of the most... Okay, so, and sorry, just in the interests of time, since we're already 1 hour in, what do you think we should do about this bacterium? Please put it in the chat so that we get through it faster. What do you think that we should do about this bacterium? One is use some antibiotic.
And so my question is, if you're using antibiotics, won't it just become like a super bug by mutating quickly like influenza?
Right. So there's always the problem of antibiotic resistance when you're having an antibiotic.
Superbugs kill like 700,000 people in America every time.
Yes. So and I agree, antibiotic resistance is a big thing. Heat. Yeah, Lokpriya, that's a good idea. But
Pesticide.
farms are very, very big.
Pesticide.
Okay, so
Pesticide.
pesticides kill pests, which are usually insects. They don't do anything about bacteria.
Oh.
The compounds that kill bacteria are called antibiotics. They should technically be called antibacterials, but that's fine. Okay, so
But,
all of
sir,
you are saying find some way.
I have
to
an
get-
answer, sir. I have a new answer.
Okay, one new answer and any other new answers, please type. Another option is, so actually the 1961 study where he was infecting those plants, I think Krishana was the one who said maybe there, or someone mentioned immunity. The reason he was actually doing those experiments was to find strains of corn that are resistant to the fungus.
Oh,
So
yeah.
maybe there are some strains of plants that are resistant to this, and that could work. Okay. So I think we should study it more. Okay. So now let's quickly go about studying it a little more. Okay. If you wanted to study this bacterium more, what questions would you ask about it? Please write it, shorten it and write it down. So someone here has mentioned genetic engineering.
Maybe grafting.
Okay. So genetic engineering of bacteria.
I think
someone also mentioned phages. I'm really glad someone here knows phages. Phages are viruses that attack bacteria. Okay. Okay. So hold on. Wait. So let's write this down. Questions. Which plants do they infect? Okay. So conditions to grow. Which plants do they infect?
What's a bacterium?
Bacterium is singular for bacteria. Like person and people.
Sir, how do the changes in environment affect mutation rates in bacteriums?
In general? Or you're saying
Yes.
that's a specific question you're interested in answering?
Yeah, it's a question I want to be answered.
How do changes in environment affect mutation rates? Okay, that's a good question. Understand the structure and help plant immunity. That's a nice idea. Help plant immunity. Okay.
Maybe
what conditions do they need to grow?
maybe like we can find maybe they can find a way to grow to mutate a seed of a plant and create a plant that thrives on the bacteria.
Okay, so some kind of resistant plant in whatever way you make it resistant.
Or something that, because it's a problem that grows because of this bacteria.
So that was, just to clarify, that's what a bacteriophage is. A bacteriophage is a virus that specifically eats that bacteria. Right? Where is it found? Yay. Okay. No. It's so I'm talking about that situation where there's a bottle of supercooled water and you put the bacteria in. Okay. If you do it with the bacteria the Pseudomonas that do not have this protein, it will not form ice. So then through genetic engineering, we know that there is one particular protein that makes the ice form. Okay. But now we finally come to Krishana's question. Why could making ice be useful for a microbe? Okay. One part we've already kind of got at, which is ice damages plants. Right?
Why do you want to damage plants?
I mean, for food. Right? Plants are, like we are all living things are bags of water with lots of food. The bag is preventing you from, or the bacteria.
Is whatever that's.
So the bag is preventing the water preventing the bacterium from getting to the food that is inside the water. If you make a sharp ice crystal that punctures the bag, now all the nice nutrients are coming out and you got food. Okay. So the other thing is, have all of you heard about igloos? Why would you live in an ice house?
Like to reduce the cold kind of thing, because snow
it insulates.
absorbs the cold.
Because the ice will keep all the cold inside of the ice and keep the air warm.
That doesn't fully compute, right? Ice is cold. Why is living in a house made of ice warmer than living outside?
Because it
Because
keeps
the ice
the warm air
traps
within
the
Wait,
cold.
hold on. Rakshad, if you're up to speak.
Yeah.
To insulate the air inside the igloo, and so that will make you warmer.
But you're surrounded by ice. Why is it making you warm?
Because it's absorbing the cold.
Because inside the ice there is air, so that air will like insulate the thing inside, like the space inside.
So I'm going to add one more thing. It's air is an insulator. Ice itself is an insulator. Right? So if the air is very cold outside, okay, and you also see this in cold places, right? The folks in Scandinavia will understand this. A lake can be fully alive during winter when the temperature outside is minus 30 degrees Celsius. Because the lake is like this, it's minus 30 Celsius outside. A layer of ice forms on top of the lake. And inside the lake, it's around 0 to 4 degrees Celsius. Right? That ice is being an insulator from the minus 30. So now the thing is that if you let ice form however it wants, such that it creates crystals that puncture your cell, that is bad for you. But if you are able to build an igloo, then that is protection from the elements, because the ice itself is an insulator. So the bacteria's ability to make ice around itself can help it make its own igloos. The other thing that we discussed already was the ice can pierce cells and other structures and release nutrients. And it also helps spread the bacteria, which we will get to in a bit.
So one more point is that surviving cold conditions, some microbes control how ice forms, so it doesn't damage them too much.
Yes, agreed. So that was the first point that we said over here. When you control how the ice forms, the ice itself acts like its own personal igloo.
It takes over space also.
Yeah. So it essentially creates. So agreed. The bacteria is still at 0 degrees Celsius. It's cold in there, but it's not dead. The ice has formed around it and it's able to continue surviving. It doesn't get exposed to the minus 30 degrees Celsius, which is outside. When I showed you this caricature of a lake in Minnesota.
Did not understand.
So if it's minus 30 degrees Celsius outside and you build yourself an igloo, it's quite possible that the temperature here could just be minus 5 degrees because the ice is insulating you from the outside. You build the igloo when the temperature was minus 5 and you stop.
The reason the outside is cold because of the ice.
No, the outside is not cold because of the ice. Your igloo is not making Antarctica cold. Antarctica is cold because of the climate. But your igloo can insulate. Dhruv, I get it that you have questions. I'm going to ask that you wait and we'll discuss this later. So, where all do we find it? On trees? On plants? We've already discussed that, right? It's obviously going to be in the oceans because it kind of evolved there, right? We said that it started out from the oceans. They also find it in the Arctic Ocean because we've been talking about ice. You take ice and ocean and that becomes Arctic Ocean, right? Here's the funny thing. People find it in alpine lakes and streams. You go into mountains that are far away from any crop fields, and you find it in the water.
Find what?
It over here is Pseudomonas syringae, the hero of our story. Okay. They find it in underground aquifers, the water that is there underground. They've also found it in the center of hailstones and in freshly fallen snow. So if a bacteria is in the center of hailstones and in freshly fallen snow, where is it coming from?
The
Krishna?
clouds.
Clouds.
The clouds.
Water,
So
because water
So you're
condenses.
telling me that the bacteria is in the clouds?
No, water.
Yes. Bacteria.
Yeah, bacteria would be in clouds, right? Because if bacteria is in water and clouds have water, then it means bacteria would be in clouds.
How are clouds formed? Clouds are different from ocean and seawater, right?
Water cycle.
No, wait, what if it was in the atmosphere already? Because water
Yeah, it was in
condenses.
the atmosphere.
So that's possible.
So an important point, clouds are made by water that evaporates. Now when water evaporates, it leaves behind everything else. Only the water evaporates. Right? Because it's just water molecules that are becoming gas. That's literally how you make salt out of salty water, right? You spread it out on a pan and let it evaporate. All the salt is left behind, the water goes up. That water that goes up, extremely pure water, goes and becomes clouds. Okay, it is literally like distillation, as Rakshat is saying. Yeah. But remember the research I told you about earlier, where they were trying to see what is the best temperature that they can get things to condense at. One of the things driving that research was people had, using airplanes, measured the temperature of clouds. And clouds are having condensed water and ice at -3 degrees Celsius. But those scientists, they tried all kinds of things, and the best that they could get with trying dust, all kinds of particles, the best temperature they could get condensation at was -5.
So how is that possible? Because pure water like crystallizes at -30, and this thing over here is.
Okay, so just to clarify, so they've measured the temperature of clouds, and the temperature of clouds with ice in it is -3. You already know the answer, like it's the point of calling it the hero of the story.
It's the thing which makes it, I don't know, cold.
It's the bacteria.
Oh.
Right? The same bacteria that's around everywhere. It just gets carried up by the wind. When you find it in the center of hailstones and in freshly fallen snow, that means it was in the cloud. If it's in the center of a hailstone is an ice crystal.
How did they
formed around the bacteria.
How do they detect it?
It's standard. You take the hailstone and culture the bacteria. You put it on a plate of good food for the bacteria.
Like, even if there's pure water, there's also some amount of salt content in the cloud because of the dust. So how is the bacteria surviving? Isn't salt condition supposed to make it?
No, so note there is a difference between surviving and growing. We are not saying that the bacteria is growing in the atmosphere. Nearly all bacteria can enter a sort of a spore state, right? A state where they're in like suspended animation and they're just waiting for conditions to be right.
Like a virus, basically. A virus just randomly touches into a cell.
Viruses are not alive during that period, right? So viruses work differently. But bacteria can sort of just hunker down and be waiting for conditions to be right. So it need not be that they're actively metabolizing up there. But all of this means, slides. All of this means that this bacterium's life cycle is the whole water cycle. And this is something that like scientists only figured out about 20 years ago. We've been studying this thing since the 1960s, but we only figured this out in the 2000s. Okay. It's there in the clouds, it's in snow, it's going into underground water, it's going into the water table, it's in rivers. It is
What's the
also
water table?
not relevant. Region of water down below, I mean just region of water down below. So it is also in crops. Now the thing that was happening was, for the first part of this story, this is how we had framed it. We were only looking at the crops and the bacteria in the crops, and therefore the answer to the problem of this bacteria causes disease was kill the bacteria. Now that we've zoomed out, what do you think would happen if we killed the bacteria?
The plants won't survive.
It won't rain, kind of thing. Less rain.
There would be less water, so the plant wouldn't survive.
The plants won't
Less
survive.
rain.
I mean, if we kill the bacteria, we're messing up something very. I don't know what would happen.
The water cycle.
Yeah, but basically, if we kill the bacteria, we're tampering with something huge. We're tampering with the water cycle. And that has unknown consequences. Right? So this diagram is from a scientific research paper published in 2014 that shows that this Pseudomonas is involved in flows at local levels, at regional levels, and at global levels. They've been able to track this through the genetics of the bacterium, that they see samples that come from Eastern Europe end up in Western Europe. It's traveling on the scale of the whole water cycle.
But why would it travel? Why can't it stay in the same area?
The water cycle is
control,
carrying.
right? It's a single cell. It's the size of a micrometer, right? 1,000th of a millimeter.
Yeah.
It doesn't necessarily have control over. Okay, I'll save that discussion for afterwards. Let me just finish the points that I was making. So just to close, because some of you might want to leave, and I see 10 people have already left. The point, one of the moral, well, if there is a moral to this story, it is that our field of view shapes both problems and solutions. When our field of view was just the field, and we think about only the crop, the solution is kill the bacteria. In the 1970s, the same scientists who figured out about ice nucleation created a genetically engineered variant of Pseudomonas that did not have the ice protein.
What happened?
They tried to do limited field trial, like they tried to do trials of it, but they were blocked by activists in the US. They did it even without, they tried to do the trials even without permission. In the 1970s, they did not know this story. This story emerged only in the 2000s. But suppose they had done widespread introduction of that genetically modified bacterium, we have no clue what it would have done to the water cycle. This is why, and I'm not saying that genetic engineering is bad, but genetic engineering done with a narrow view of only wanting to solve the problem of the field without seeing what is that this bacteria is the whole water cycle. That was definitely a bad idea. It's good that those trials did not happen, and the frost-free version was not sort of popularized throughout the world. Pseudomonas is still a big problem for agriculture. It causes billions of dollars of loss every single year. But the answer isn't going to be something simple like put an antibiotic. The answer might be grow more diverse crops. If you have a field with many crops growing on it, then a single bacterium that's a problem for a single crop doesn't take over the whole field. Like,
that's what most people do in
Sir,
vaccination. Not everybody is vaccinated, but enough people are vaccinated so they can't get affected and the other people won't get affected too. That
right.
means
So
even if one plant gets affected, the others won't be affected.
vaccination dynamics is part of it. The other is just more diverse agriculture, right? If you have hundreds of fields, so since we started with corn, let's go with corn. Corn was domesticated in South and Middle America. Okay, in Mexico, every single valley has its own variety of corn. Just like rice in India, there are salt-tolerant varieties of rice that are used in Nagaland, in Odisha, that scientists in the Philippines have spent millions of dollars trying to develop when that rice variety already exists in Odisha. So there's a lot of biodiversity out there. But when the whole of the United States is growing 3 varieties of corn, a fungus that is suddenly successful on one variety of corn can decimate the whole production of corn in the United States. But if they were growing hundreds of varieties of corn, that wouldn't be a problem, because because each of them would have slightly different immunity and therefore work differently. Okay. I'll send this video later. Pseudomonas is actually inactivated versions of Pseudomonas are used to make ice for ski resorts, for all kinds of human applications where ice is not being consumed. Right? They use this bacterium because it is the most effective ice-nucleator known. Just a point of how the ice nucleation happens, and then I will close up. Okay, also, those of you who are still here, we have a couple of programs upcoming, including next week. So I'm just putting it in the chat. So next week and the week after, next week, we have a parent hour of what does it mean to learn from YouTube. It's for your parents. Please tell your parents. Okay. And then we have another career session coming up 2 weeks from now, which is a which detail is in the chat. So, okay, let's go with something easier to think about. Okay, let's just go with humans. Okay, an antibiotic is something that kills, or an antibacterial is something that kills a human. Kills a bacteria. What are ways that you would kill a human?
Sir, you would drown them or stab them with a knife.
Stab them with a knife. Okay. So someone's going around stabbing humans with a knife. You are a human. You are able to. So over evolutionary time is what happens with bacteria. But now let's just say humans are deciding things. How would you protect yourself from being stabbed with a knife?
Sir, I would run away, or I would try to block it with my hands, or I'd wear some armor or clothes that protect me.
Okay, you could wear armor or you could run away. Cool. Once you wear armor, does it protect you from certain other implements other than knives?
no.
Yes, sir. Other things, it can also protect you. They may not be as sharp and they may be similar, but since it's the same technical, it's just the same thing that's hitting you again and again, so it might.
Right. So the same armor could protect you from a knife, from an arrow, from, I don't know, a flanged implement like a mace. Right? So the exact same thing is happening with bacteria, except that instead of a human deciding to wear things, it is random mutation and evolution that's causing these changes. Suppose the antibiotic is something that pierces the bacterial cell membrane. And therefore spills it out. The bacteria that develops resistance creates something that goes out and blocks the piercing. You can have you can have antibiotics that go into the cell and mess up cell processes. Resistance can be by creating a pump in the cell membrane that is chucking things out. Right? Stuff that it recognizes as foreign or not belonging to the cell gets chucked out of the cell. So there are a whole range of classes of there are different classes of antibiotics. So just like we said that the armor will protect you against arrows and knives and maces, right? There's a similar thing that it develops some particular kind of armor and it protects against a certain class of antibiotics, and others might work. So the thing with bacteria is they evolve very fast. The next generation comes very fast. They mutate at a high rate. The more mutations there are, the more different protections that get explored.
Yes, sir. And my second question is that why do some bacteria develop resistance very quickly while others exposed to the same conditions do not?
So that as I said, is a mix